close your ears. Once in a lifetime, you get a student that does everything that you expect, plus 200% more, and fast. When I met Lena last year, I was incredibly impressed by her background, by her dedication to work that she does, and by combination of interest in the subject and the ability to do things fast. The model that they put together is probably one of the most complicated models that we will see in all those seminars. Nevertheless, it only took, what, 6 months to put it all together, and now it is in the testing phase. So, with great pleasure, I introduce Lena and her model.
Thank you to Anatoly for that introduction. I'm Lena Sherbakov. I'm a second-year GPN student, and I will be presenting on ongoing work that I've been doing with the Neuromorphics laboratory around a visual model that we call CogEye. So the goal of this talk is really to give an intuitive sense for the type of modeling systems that we're building, and not to dive into the mathematical detail of the models. But if that is a question about the presentation, I would be happy to address those after the talk. And also to highlight the potential usefulness of such a system. As Anatoly mentioned, it is complex, and one of the fundamental questions we always ask ourselves is, does it need to be this complex? And what is the advantage of having such a system? Okay, so what is CogEye? So CogEye, as I mentioned, is a computational model. It's centered around a biologically inspired visual system, and it runs on HP's platform, CogEye Machina. So I'll talk a little bit more about the software platform that CogEye runs on, but what the visual system is meant to replicate are several visual areas. Processing from the retina through the LGN to the where and what pathways of the visual system. So we're both trying to simulate how a person makes the saccades that he makes, what governs those saccades, and then what happens in the what stream. So how do we bind different views of an object to a consistent object category, and how we make sense of what we see in general. So here's a brief video about showing the types of saccades. So this is the where system in our model, making these foveations. Where do people look when they're watching a tennis match? Can turn off the volume. This is a heat map of where people tend to look. This is already some object parcellation of what that heat map is representing, and this is just a look-through view. But these are the types of saccades that we're hoping the where system could learn to make. And the what system that I mentioned earlier is what makes sense of what it's seeing. Okay. So a bit about the platform. Why is it called CogEye? Well, it's called CogEye because we are modeling this on HP's platform, CogEye Machina. HP and BU have had this collaboration for the past 3 years, where we're developing a neural modeling platform that runs on GPU clusters. And the idea there is we can take advantage of the parallel processing architecture to build these models that can process in parallel instead of sequential processing. Another advantage of Cog is that it has a built-in virtual environment, so we can test these models on any type of virtual world that we build with any type of animats. So if we want to build a virtual rat and test to see how the eye in the rat saccades and looks at these scenes, we can. Another big advantage of the platform is there's a lot of work around integrating it with robotic platforms. So we can eventually put these models on a robotic platform or on a mobile platform. Okay, so the goals of CogEye, I've broken them down. These are major goals of CogEye. There's sub-goals in each of these. But the first is space variant active vision. And what I mean by space variant is that when we sample a scene, the retina doesn't have resolution, the same resolution throughout, obviously. The fovea has much higher resolution than the periphery of the retina, and we want to replicate the same thing with our model. And I'll talk about some of the advantages of doing this. Why would we even want to have this space variant sampling? Active refers to the idea that CogEye is not fed where to look at all. So nobody is telling it, look here or look there. It must decide on its own, on each saccade, where it's going to look. Classification of views, so each time it sees something, it must make sense of its own, must classify those views somehow by their similarity. Binding of views with objects. So with every single view that we acquire, a very fundamental question to any visual system that we often take for granted is, how do we know that those views belong to the same object? So even if the object is stationary and our focus of foveation is slightly different, we're going to get a very different representation of the object. And how do we bind all of those different views into a consistent object representation? And lastly here, binding of spatial location with object evidence. And I'll talk a bit more about this because it's not too obvious from the beginning what I'm referring to. But the idea is that if I'm gathering evidence and I'm pretty sure that I'm looking at an object, say a 0, I'm going to have some foveations that are more informative than others. So not all foveations carry the same amount of information. So the idea here is to build up this evidence map of, if I'm fairly sure that I'm looking at this object, where should I saccade to make the most of that eye movement? And lastly, to provide a global framework for moving forward. So we want this visual system to integrate with other systems that we're building, such as navigation and path planning. Okay, so why do we need it? I've talked about this space variant vision idea. Why do you even want to replicate the fovea and the retina in general? The idea is to process an image in its entirety with the level of detail that a fovea would, the level of resolution that a fovea samples at, the amount of resources, both material resources, spatial resources, energy resources, is extraordinary. For example, the fovea is only 1% of the retina spatially, but occupies more than 50% of the visual cortex and more than 50% of the neural fibers running from the retina are foveal. So if you expand this 100 times, you can imagine a computational explosion of the amount of wires you would need, the amount of neurons you would need to model everything at such a high resolution. Energy consumption is another big reason, and probably the biggest is speed of processing. So if you're in a situation where you must act very quickly, and you can either make one saccade to determine what is the danger in the situation, or you can spend the time resolving everything in the same amount of detail across the entire scene, you're much better off with the option of making one saccade to immediately resolve what the danger in the situation is. And other reasons, really, to have a system that learns as opposed to having a hardwired memory bank of memories is really, if you don't know what you're looking for in an environment, you're much better off with a system that can learn to look for certain things as opposed to searching through a memory bank of pre-existing experiences. So these are some of the reasons, and I can go back to some of these if we have time at the end of the talk. Okay. So high-level diagram of CogEye. So as I mentioned,
Lena, can I ask a short question? You mentioned saccades. What is the frequency of the saccades?
Yeah, it's roughly 3 saccades per second.
Okay.
Yeah.
Thank
So
you.
we're really we're not modeling microsaccades or the smooth pursuit.
Okay. Thank you.
So the early visual system in CogEye is comprised of a foveal sensor, a method for edge extraction, or neural model for edge extraction, and neural model for surface property extraction. And the surface property extraction right now is just luminosity, but will in the future include things like color and texture as well. We also plan to include figure ground segmentation and optic flow as part of this early visual area processing. Okay, so after the input has been processed by the early visual areas, it goes, I'll talk about the WARE system first. So the WARE system has 5 main goals. The first is to select a hotspot. What this means is select where it's going to look next. This is based both on bottom-up saliency, so the features that it extracts from the early visual system, and top-down feedback from the WHAT system that tells it what spatial locations are the most informative in this scene. And evidence for this type of hotspot selection most recently are in papers from the Ballard lab that show that when one analyzes a scene that's static, the saccades are very different than when that same picture scene is part of a dynamic context. The idea is the bottom up and the top down equally contribute to where the person makes their next saccade. The WARE system also is responsible for making that movement, and this is not as obvious as it seems, so it must calculate where it looks, but also make that movement correctly. It forms an attentional shroud that binds views with objects. So this is possibly the most contentious part of the model. The idea here being that the way our system knows which views belong to a single object versus a different object is that the WARE system forms a surface-fitting shroud around the foveated object to help the WHAT system classify those views as pertaining to one object versus another object. And I'll give some biological evidence for this idea of a shroud, but it's really critical to how we solve the binding problem in this model. And this isn't to say that the binding problem is solved. This is just a possible way to do it. So the next part of what the WARE system does is once it forms the shroud around the object, the future saccade that it makes can then either fall on the surface of the shroud or fall off of the surface of that shroud. And that's how we know whether we have foveated on a new object or if we're still on the old object. And if we are on the new object, the WARE system sends a non-specific blast of activity to the WHAT system. And we call this a reset signal, basically telling the WHAT system, stop categorizing for this particular object. There's evidence now that you are on a new object, start categorizing for the new object. So this is the reset signal. And lastly, inhibition of return. And what this is a working memory type mechanism for remembering the locations of where you've previously foveated. So you don't want the eye to get stuck on looking at the same spot over and over again. You need some mechanism by which you have this inhibition of return. So, and I can, if we have time, I can go over some of the papers that support these ideas of WARE system processing. So what does the WARE system pass to the WHAT system? Well, two things, the shroud modified view and the reset signal. And the reason I say shroud modified view is if you think of the shroud as covering, it's a form-fitting structure that covers the surface of the object. It has the property that it can also suppress the surround. So the fovea has this nice property of enhancing things in the center, but the surround is still present a little bit. And you don't want that to be going into what you're classifying as a view because the surround has nothing to do with the object you're looking at. So the shroud is what helps suppress the surround, and this reset signal that will tell which view categories belong to which objects. Okay, so the WHAT system, I should say right now that the WARE system was developed by Genna Leavitz and Aisha Sohail in a lab, and I designed and developed the WHAT system. So every all the credit for the WERE system. It's not mine. So the WET system is a 3-tier classifying system. The first is it must look at the views that it sees and classify those views in an unsupervised manner, just based on their similarity and dissimilarity. The second is object classification. Just take those views and group them into object categories. And the third is name classification. So take those objects, group them into names. And evidence for this type of view variant and view invariant process, going from posterior to anterior IT, was presented by several people. fMRI experiments they can also talk about after the presentation. And a big part of the WET system is also to send what we are calling a spatial evidence map to the WERE system. So when I accumulate information about where the important foveations are based on how I'm classifying them, I can feed that back to the WERE system as top-down input for the hotspot selection. So now it's not only selecting where to look based on bottom-up saliency, but also based on top-down knowledge from the WET system. Okay, so the WET system can also take as input an external teaching signal, so something that tells it what the names are of the categories that it's looking at. This helps in the initial stages of learning names to objects at that time. And lastly, what the WET system passes back to the WERE system is a spatial evidence map that helps the next selection of a hotspot. And importantly, this work was inspired by a paper, several papers by Fazil, Foley, Grossberg, and Bengola. The model was called ArtScan. A lot of the ideas, the computational ideas, are similar. The implementation and the algorithms are very different, as well as many of the additions of the spatial evidence map and so forth are different. But I wanted to acknowledge ArtScan here. Okay, so now I'm going to go through the several different modules of CogEye in a little bit more detail, starting with the early visual system. So what the early visual system does is replicate what the retina and the cortical magnification that you see. How we model this is with a log polar transform of the input image. So if you're looking at a sea of digits and you're foveating on the 8, this is what the scene would look like. And the early visual system will also extract the surface properties and the edge properties from that image. So if you're looking at the 8, these would be the edges that it extracts. We also hope to expand the early visual system to include color, texture, depth segregation, and optic flow. Okay, so the WERE pathway. This is the system diagram. I'm not going to go into the details because it would take too long, but the idea here is all of the round oval circles represent part of the processing of the WERE pathway that is done in eye-centric log polar coordinates. The squares are all of the processing that is done in a different coordinate system. So why do we need all of these different coordinate systems and to constantly have to transfer between coordinate systems? The idea is that if we're acquiring the image in this foveal log polar eye-centric view, how do we give the eye the instruction to move some linear distance in pitch and some linear distance in yaw in these log polar coordinates? Right? So you have to make this transformation somewhere. And this is a lot of what the WET system does, and not as simple as just saying move your eye here, move your eye there. Okay, so rather than go into the mathematical details, I want to start by looking at some of the examples. An added benefit of working with COG is that every neural field that you create is probable. So you can click on it and look inside and see what the processing is looking like, what is going on in these fields of neurons that we're programming. So here we see a two-dimensional JPEG. CogEye is instructed to pick its foveation points and run on this 2D image. So all of the pink spots are places where it has foveated before. Here you see the raw sensor. So this is the retinal sensor. This is the shroud in the process of being formed around the object. This is the shroud once it's stabilized and what is sent to the wet pathway. And these are selections for the next hotspot. So I'm going to run this now. Okay. So you can see right now we're focusing on the 0. Now we've switched to the 5. And as the shroud forms, you can see how it fills out and is able to form and then suppress all of the surround that we don't care about because it's not part of the object. This is really nice because the wet system can now classify a cleaned up version of the image. We don't want to be classifying this, it's got all of this surround. And the reset signal is a binary that gets pulsed whenever the object changes. And this is again a computation that is done. It's not as simple as turning a switch on and off. I'll go into how that's done. This is the yaw and pitch. So these are the instructions for the eye, how much to move and where. And these are, the little dots are the hotspot, next hotspot locations in different coordinate systems. Okay. So the wet system, as I've described, is a 3-tier classifier. The first being an unsupervised view classifier, modeled as an adaptive resonance theory network. The object category layer is a recurrent competitive field that where the neurons compete and accumulate evidence for what object they think they're looking at. And the name classifier similarly is a recurrent competitive field that accumulates evidence. Once the object category system has accumulated enough evidence, it can send information to the WARE system and say that based on what you're looking at, I think the next spatial location that would be very informative for you would be such and such. So that's the type of information that the WET system passes back. Okay, so an example of the WET system processing. So I'll play this movie now. Oops. There it is. Okay, so again we're looking at a sea of digits. Here you will see the view category cells that are activated by a certain input. Here are the object category cells that are activated, and here are the name category cells. And I should say that these are evidence accumulating cells. So this is really the amount of evidence for the object, the amount of evidence for the name, as opposed to a direct output category. So you'll see as a certain digit pops up, some percent of the view category cells will be activated. So this 7 activates this field of cells, which activates this field of objects, and this field of name categories. These are the synaptic weights connecting view to object, and similarly we have synaptic weights connecting object to name. Here you'll see what I'm calling the spatial evidence maps. So as it saccades around a certain object that it knows its name, it will accumulate evidence of what foveations were important for me. So if I have a lot of evidence that I gained by foveating here on the 0, it's going to be a much brighter spatial spot for that object. And so this is 0, 1, 2, 3, 4, 5, 6, 7, 8, 9. You can see that it starts to form these, it hasn't run for very long, but it starts to form these representations, size invariant representations of spatial location based on object. Yeah.
Is this supervised or unsupervised here?
So yeah, the view classifying is completely unsupervised.
Okay, so you're not telling it where to look?
Not telling it where to look.
Sorry.
Yeah, nothing. The object category, it's a recurrent competitive field that it's supervised in the sense that the where signal tells it what to group. So when I get a blast that I'm not looking at the same object anymore, I'll reset my recurrent competitive field. And the same for the name category. So these spatial maps are really informative because once we have an idea for the spatial evidence of a certain object, if you're looking at a 5, and for example, you're not sure if it's a 5 or a 0, you can use these heat maps to determine where is my most informative saccade going to be, right? So in this case, these two locations are going to be very informative saccades if you're not sure if it's a 5 or 0, because if it's a 0, there's a lot of evidence here. There's a big evidence difference in this location between the 0 and the 5, and similarly in this location. So this is something that the what system feeds back to the where system. Yeah?
Can you go back to the previous slide?
Yes.
Yeah, so just to clarify, I see like several 8s here. So what if I make one of these 8s much bigger? Are you saying that you are going to be, the system is going to classify both of them as the same, even though it's on the same screen, one is much bigger, they are at 2 different spots, they would be categorized as the same object?
Yes,
right.
They would be categorized as the same object. Yeah, it is view invariant, and these maps are also view invariant, right? So, I'm sorry, not view invariant, size invariant. So, regardless of what the size of the 0 is, it's going to build a map relative to its size, relative to the top and bottom locations. Okay. Okay, so system validation. We validated the system in many different ways. The first is just to look at the what system alone and validate it against a very standard data set, the MEAS data set. This is the handwritten digit data set. We get an accuracy of 92%, which we're actually very pleased with because there's no tweaking or fine-tuning of parameters, and the goal of our system is not to recognize digits. It's really to show that this type of where-what interaction works, and that you can integrate the two systems. So we're also testing it currently on the integrated system, where it's looking at many JPEGs of those digits, and similarly being trained, so providing a name label, and then when it's unsupervised and has no name label, seeing how often it accurately predicts the digit that it's looking at. Okay, so I wanted to show this last because this gives you a good idea of what the system does not in an artificial digit setting. So here's a scene of baseball players. This is the actual foveal sensor, so this is what the retina sees. This is the shroud modified image once you suppress the surround. Here are all of the view categories that it's learning. The pink circles are again where you have saccaded to in the past. What this field is, it's a representation of everything that is sparked in memory by a given view. So if I see this view right here, I should not only spark in my memory the last time I saw that particular view, but I should also remember what object it's tied to and what name it's tied to. So because we have that 3-tier classification, I can search in my memory and be able to associate all of the different views of that object and not just that single view. So this is what we call a map field. And the idea is that once the scene changes, and it's going to change in a second to people playing baseball in the snow, CogEye doesn't stop. It doesn't have these learn, don't learn type of rules. It runs continuously, and you can change the scene and not tweak any more parameters, and it's going to saccade again. It saccades to the ball, and it immediately recognizes all of the views that were associated with the ball that it's previously learned, even though this is something completely different that it hasn't seen before. Yeah?
So is the map field kind of like a new teacher that is being given to CogEye, so like a new set of parameters to know for future for future recognition?
No, it's actually just an illustration of what is sparked in memory when it sees a view. So usually when I see a view, I don't just spark in my memory a single similar view that I've seen before. I spark in my memory all of the different views of that object.
Okay.
So that's what it is. It's just to highlight everything that's associated with that particular view. So I'm running out of time, so I'm gonna skip to the punchline. So the idea of combining the where and what system. What's the advantage? Well, we really think that this would be really neat to do context identification. The idea being that we're not just doing object recognition, and we're not just doing saccades. We're combining both to be able to identify objects. So, for example, a sink is present in both a kitchen and a bathroom. So a system that does object recognition would not be able to tell you where you're standing. But because we have these spatial relations between where the objects are found, what they're next to, and so forth, you can then disambiguate where you're standing based on this combined knowledge of what and where. And by this, identify the scenes that you are currently viewing. And this is the plan for what we're starting to test right now, is looking at different rooms in the house and seeing if CogEye can identify the scenes. Where are you right now? Okay, so I'm 1 minute, I'm almost done. The limitations. One is, how far removed are we from biology? So there's a lot of contention among experts around the shroud idea. Do we really form-fitting shrouds around an object? It's a very valid concern. It's just one of the ways that you can solve the binding problem. And it's a valid concern. So another limitation is that very few people are currently combining a where and what system, which affords us no good standard benchmarks to use to validate our system. We don't want to just do object recognition because that's only half of the system. And we don't want to just do visual search because that's only half of the system. So what do you validate it against with this combined where-what pathway? And our idea is context, right? You want to see if they can grasp context. Another big limitation is right now the shroud only forms in a static scene. So if you're looking at a dynamic scene, how do you have a moving shroud that captures the object? So future work, I've touched on all of this, but to beef up the early visual system, we want to include color, we want to include texture, to integrate optic flow. Something I really want to do for my thesis work is to include temporal sequence learning. So instead of just learning objects, I want to be able to learn sequences of objects and sequences of events. So this is where I'm hoping to take the system. And lastly, to integrate it with all of the other work going on in the Neuromorphics lab around navigation and so forth. So with that, I'd like to thank first my PIs, Max Versace and Anatoly Kochechnikov, but also all of the lab members at the Neuromorphics lab, particularly Gana and Aisha, who developed the WARE system, without which CogEye wouldn't work. And our HP collaborators, particularly Greg Schneider, who is the architect for COG. And we had a summer intern work on porting COG to a robotic platform, and she did a great job in starting that. And this is funding agencies for the Neuromorphics lab and personal funding for me. So with that, I'd like to see if anybody has any questions.
Probably just take 2 questions.
Okay. Yeah.
If these smash digits together, I guess, can a shroud deal with that? So it fits around the surface, if I understand it correctly. So if you have overlapping surfaces, does the shroud just continue to grow like a blob if anything is connected?
Yeah. Yeah, so the idea is with the figure ground segmentation as a pre-processing step of the early visual system, we hope to, and disocclusion as well.
Right.
We hope to get around that, but currently we don't have that there. So yes, yeah, we need some spatial separation for the shroud to form.
Yeah, I have one question. Once you finish one of the learning,
and how do you select the new hotspot? I mean, from the prefill.
Yeah, so if you don't have any of this spatial evidence top-down map governing where you're looking, because you know what the object is, then it's entirely driven by bottom-up salience. So the luminosity right now is a critical feature. So the next bright spot would be selected as a hot spot.
So there will be a competition among...
There would be a competition among all of everything in your periphery and in your foveal vision, or in your retinal vision, for that next hot spot.